So now that we've talked about major biotechnology techniques, we're in a great position to look at an example of how we've been using those techniques in the year 2020 to diagnose people that have an active COVID-19 infection. So what I've drawn so far is just a really brief cartoon that sort of summarizes some of the infection dynamics for COVID-19. So COVID-19 is a type of virus that is surrounded by a lipid membrane. And you may have heard about a type of protein that sticks out through that lipid membrane called a spike protein. And the spike protein that's on SARS-CoV-2 is one that matches up with a receptor protein type found on many types of cells in our body called the ACE2 receptor type. That's a major receptor type. So basically, if these COVID-19 virus particles are floating around and they happen to bump into a cell type that has some of these ACE2 receptors, they can bind to those receptors, and then that triggers conditions that facilitate the release of RNA that's present in the SARS-CoV-2 virus. So that RNA winds up getting released into the host cell. So this is another type of virus that contains RNA instead of containing DNA. It's a little different from the retroviruses that I talked about earlier. So it doesn't use quite the same processes as a retrovirus does. It doesn't go all the way back to DNA. But like a retrovirus, the particle that actually gets distributed around is RNA rather than DNA. So this RNA gets released into a cell. And at that point, then the ribosomes that are present in that cell will start translating that RNA, not knowing any better. So they'll translate that RNA and they'll start to then produce proteins from that virus. And the proteins that get produced are capable of then making another set of RNA that's used as a template to make RNA and proteins that are eventually going to get exported as new viruses. So how do we actually determine that somebody has a SARS-CoV-2 infection? Well, we need to be able to detect the presence of one of the substances that's associated with an active virus. So the major target that most tests rely on is detecting quantifiable levels of functional viral RNA. A little later on, after someone has had an infection and their immune system has had time to respond, their immune system will start produce antibodies against the SARS-CoV-2 virus. So you can tell if someone has had an infection recently if you are able to detect and quantify antibodies from their blood, but they're not going to have those antibodies earlier on in the infection, which is why it's really important to be able to detect functional SARS-CoV-2 RNA when it's present. So this is, again, going to rely on some of the processes I've already talked about. So in order to detect it, the first thing that has to happen is we have to go from RNA back to DNA, right? Because we have a lot of ways of sequencing DNA that's present. So this relies on using reverse transcriptase. Again, okay? So once we have that RNA reverse transcribed back to DNA, then we can use all the tools at our disposal for sequencing DNA that's present. So what we'll do is we will develop a set of primers that are specific to the SARS-CoV-2 DNA that we've generated. So they will only amplify that particular DNA sequence. And of course, in order to get there, we also had to back up and just sequence the SARS-CoV-2 DNA most likely using next generation sequencing, probably a combination of different types of sequencing, just to be sure that we know a sequence that's unique to functional SARS-CoV-2. So then we can develop primers that are specific to the SARS-CoV-2 DNA, and then we can employ the process of PCR to amplify that DNA if it's present. So if SARS-CoV-2 DNA is present, then if we run through the polymerase chain reaction process, what will happen is normally we're going to run somewhere around 40 cycles of that process. So if someone has an active infection, those primers are going to bind to that sequence of DNA that we've created, and they're going to start amplifying that sequence. And over time, over repeated rounds of PCR, we'll start to see a buildup of that particular DNA product. So if someone is positive, what people have actually started doing is using an amplification process that creates a fluorescently labeled product. So you'll see a buildup in fluorescence if there is an active infection. And if someone does not have an infection, in contrast, there's nothing for those primers to bind to, so you won't see that same type of buildup of that DNA sequence. So the process of PCR typically, so that involves heating up DNA, causing it to denature, cooling it back down again to give primers time to anneal, giving that time to elongate through the action of DNA polymerase III, then repeating that entire process about 40 times. We've gotten pretty good at very quickly heating up and cooling down little tubes full of substances, so it doesn't take us days to do this, but it still takes a couple of hours to go from the stage where you have a sample from somebody to the point where you have gone through enough rounds of amplification that you can distinguish a case where someone actually has active SARS-CoV-2 from cases where somebody does not. And hopefully you can also appreciate from this that the logistics involved in collecting these samples are a big component of what can take a long time. And then getting together all the ingredients, having the right lab, having the right instrumentation to actually quantify this also can wind up involving a lot of logistics. But hopefully now you have a good understanding of how those cases can get very reliably diagnosed. And this process is very sensitive. So you really only need a couple of pieces of viral RNA in order to be able to reverse transcribe it and then detect it using this method. So it's a very, very sensitive method. But on the flip side, it takes a lot of time and a lot of resources to run these diagnostic tests. So we're limited in our ability to really ramp up this process and test everybody frequently using it. And so in the meantime, there are some other components of the virus that can also be detected using other methods that aren't quite as specific or sensitive, but that can at least give us some idea about how widespread SARS-CoV-2 infections are in any given group of people.